Okay. All right. Well, welcome to Tuesday evening lecture series. This presentation is being recorded, and once processed, it will be uploaded to our YouTube station channel for anyone to access at a later date. And I think a few more people joining in. We will do a question and answer session at the end of the presentation, but you're welcome to type in your questions in the chat window, and I will try and relay the question to the speaker at an appropriate moment. Tonight's lecture is Rhythms of the Night, using bats as monitors of pollution in Baltimore City, and it's being presented by Christopher Blume. Christopher is a graduate student at Virginia Tech in the Department of Fish and Wildlife Conservation. He previously did his graduate research work in Baltimore as part of UMBC's Geography and Environmental Systems Department, and was part of the first cohort of UMBC's Eye Care program, which looks to bring diversity and inclusion into the environmental sector. Chris swears that Maryland is better than Virginia. Just kidding. Well, with that, Christopher, you have the stage.
Awesome. Thank you so much. Thank you for the introduction, Neville. Let me see if I can get my screen all set up. Cool. Can everyone see that?
Yes.
Put this
I can.
into from beginning. All right. Well, thank you everyone that came out tonight. Thanks. This is a great opportunity for me. It's been a while since I really got to talk about this work since I finished my time at UMBC. So it's really cool to kind of go back through my files and all the documents and kind of revisit what exactly I did, because it was a big part of my life for about 2 years. Not that it's gone. It's really cool. It was a really fun project to do. So I'm glad to talk about it. Let me make sure I got everything. Okay, cool. So my project, as it says, was using bats as biomonitors of heavy metal distribution around the city. I'll provide a little bit of context to the problem itself, as well as why I decided on using bats. Why are we talking about bats when this is the Baltimore Birding Club? Yeah, I'll provide all those details in a second. So first, I want to start off a little bit about me. So who I am, how did I get to do this kind of research? For a while, I didn't think it was possible. I didn't know that anyone studies wildlife or bats for a living. So I started off at Rider University, which is a small university up in New Jersey, not far from where I used to live. I studied bio there. I liked it a lot. But bio is pretty broad, and I always knew I wanted to do wildlife research or some kind of animal work. So after I left Rider, I went and I became a vet tech, and I was a vet tech for a couple years. First, I started off with small animals like cats and dogs and rabbits. Eventually moved over to a different position where I was working with larger animals like monkeys and some cows and horses, which is really neat. But still, I wanted that wildlife experience, something about working in a lab. While neat wasn't exactly my calling. So my next step was I went to University of Delaware, and I started working there in their wildlife conservation program. And I got to do a lot of hands-on work, and that really kind of set me on the path I am now. I did some research there with salt marsh and seaside sparrows. That's a little bit more of like what you guys know and work with. I was doing counts on Chicoteague Island with them, looking at them because they're very much threatened because of sea level rise. So I got to look at them and fecundity and see how they're doing, which isn't great, but it was a really cool experience. After that, I got into UMBC, and I was, as Neville said, I was in the Department of Geography and Environmental Systems, as well as the Eye Care program. The basic the general gist of the Eye Care program is to bring diversity and inclusion into the environmental sector. There's not so much of that, so it was a really cool opportunity for me to get my hands on, get in this field and kind of, get a foot in the door. And that's how I discovered bats. And that's how I discovered this project and the work I did. And it was really rewarding, really a great experience for me overall. So now that brings me to where I'm at now. I'm at Virginia Tech working on my PhD, and I am still working with bats. I'm going to explain why. I think they're awesome creatures. I think they're really great, especially working with them in an urban area. You can learn so much about not only their health and ecosystem health, but also just, I guess, general health of our systems from working with them. So it's been really great. So when I came up with this project in Baltimore, the first thing I wanted to look at, I was interested in pollution and how pollution impacts humans as well as the wildlife, since that's my background. So heavy metal pollutions are these toxins that come in relatively small doses, but can have great impact on humans' health. They bioaccumulate over time, which I'll explain a little bit more, but that just means that they build up in human systems over a period of time. The ones that I wanted to look at in particular were arsenic, cadmium, chromium, lead, and mercury. Those are very prevalent, especially in heavily industrialized areas. So places with industry and traffic, they can get into groundwater, they can get into the soil. So I thought that was a great place to start with Baltimore, that has a very big history of industrialism and industry. Also, there's an environmental justice component to this. So urban farms and I noticed a lot of urban farms when I was moving into Baltimore, and they come from places that maybe the buildings had been torn down before or had been abandoned and vacant and been demolished. And then all these farms grow up, which is a great thing to happen. But the possibility that some of these contaminants could go from the buildings, the demolition into the soil and impact the humans that are now trying to be fed or feeding people, I thought was a very interesting interaction between an urban problem and wildlife as well. So again, these heavy metals and the pollutants, they damage our health as well as the wildlife ecosystem and their health, because they accumulate and biomagnify. So I thought that was a great place to start. To continue on with environmental justice, I wanted to bring up this map. So this map shows the redlining of Baltimore that had happened in the 1930s. So basically, the homeowner association had basically sectioned off different parts of the city and gave them a color code. So green and blue parts were more favorable. That's how they were kind of sectioned. Yellow, a little less favorable, and then red was seen as like the worst parts of the city, parts that like people wouldn't want to live into. But some communities did end up living there. So black, brown communities of color, they ended up in these situations, as well as a lot of the industrial, the companies. It was a cheap area that they can build into. They could do the work that they needed to without having any kind of connection to the people that lived in that city. So we see a lot of the health problems and a lot of the contaminants that we see are normally in these redlined areas. So that was also a part of the problem or the issue that I wanted to tackle. But I also want to use the lens of wildlife as a way of doing it.
Hey, Chris, what year is this map from?
This map, man, I forget where I got it from. Actually, I might have it up here. This map might be from the Census Bureau. I might have taken it off of their website when I was coming through for the research. I can put the link on, and if anyone wants to check it out as well. No problem. So as I was developing this research, these are the 4 different topics or my goals that I wanted to tackle. So I wanted to look at the movement of heavy metal pollution around the city of Baltimore, as well as Baltimore County, just to see where they were and where they could be getting into. I wanted to examine to see what the levels of these pollutants were, and if it was at a level that could be impacting the people there, as well as the wildlife. I also wanted to connect with community organizations about this issue so that they can kind of guide me as well. I'm not from Baltimore, so I wanted to get their perspective on the issue because I felt it was right. And then also, since my background is in environmental work and I'm interested in wildlife, I also wanted to kind of bring like environmental education and maybe some community stewardship, get people involved in the process of rebuilding or taking back away from these contaminants in these urban areas. So those were the 4 goals that I had as I was coming up with this project. Some terms I just want to go over, so I'm not saying anything that's over anybody's head. So bioaccumulation is a big part of my project. Again, that's when substances, or in this case, contaminants, are absorbed into an organism faster than they can be excreted. And we see that a lot with the heavy metals that I was studying. Biomagnification is an increase in the concentration or the tissue of these organisms. So as they bioaccumulate, they build up and they magnify. And once they magnify in the tissues or the organs of these wildlife or humans, they have worse and worse complications that come with it. And then finally, biomonitors are organisms that can accumulate these toxins, and then you can use those organisms as a way to look at the health of an ecosystem in general. So again, a little bit more about biomonitors. So they're species that give us information about the health about our ecosystem. Mammals are oftentimes used, and there's two ways of going about it. You could go getting invasive samples, which is more of going in and getting a tissue sample or getting a blood sample or an organ sample, or you can go non-invasive. But either way, it gives you information about what these species are coming in contact with, and you can use that to learn more about what we might be coming in contact with. Normally, so for my project, and I'll get into it a little bit more, I want it to be non-invasive about my sampling methods, because bats are already a species that are kind of in trouble in urban areas or on the decline, and I'll get more into that. But normally, I think a lot of studies I've seen have used invasive methods for tissues, but I go a little bit non-invasive with mine. And then here are some other species, just so you know, it's not just all mammals. So we got things like the caddisfly, which are often used as biomonitors for aquatic systems. And then mushrooms are just great in general. They're great for bio remediation to help clean if there are heavy metals. And they can also be sampled as a bioindicator or biomonitor, so we can learn more about what pollutants or contaminants we're seeing in the system. But we are here about bats. So why am I using bats as a biomonitor? So I chose them mostly, it was scientific, but also perfect. I mean, my personal choice. So bats make great biomonitors because they're urban adapters, which means that they're species that have changed their behavior in a way to live next to humans. Some of that behavior could be foraging. So one of my favorite examples is that bats will go to like lights, and they'll use that to forage instead of going out and trying to find moths and insects that they normally eat. So they've adapted their behavior in foraging and roosting as well. Also, there's a lot of species that are very common in urban areas. One of them is the big brown bat. So even though it might be difficult to find them, they are there. So there were species I wanted to do. They also have a high trophic level, and their diet is made up mostly of insects. So trophic level means that they're very high up on the food web or the food pyramid. So they eat a lot of insects as well. And all those insects that they eat, that contaminants that the insects come in contact with, then move into the bats. And then I can then sample from the bats to see what contaminants will come in contact with. Also, even though there are a lot of bat species in urban areas, there's not a lot known about them and their behaviors. We know some things, but we don't know everything. They've also been declining in recent years due to urbanization as well as disease. So I wanted to study them more to see if I can learn something more about them. Let's see, what's this? So I'm just going to move this over. Okay. So a couple of things about bat ecology or Baltimore bat ecology. So bats are actually the second largest mammal order. They have almost over 14,000 species. The most abundant or the largest mammal order is rodents. So bats are right behind them. So there's two different types. There's microcryoptera, which are the species that we have here. And those microbats are basically ones that use insects, echolocation to find their prey, and then they eat insects. Those are all the bats that we have here. There are other bats that are megacryoptera. And what that means is that they usually eat like fruits. They don't really eat insects, but all the bats we have in Baltimore eat insects. They have a summer roost, a location that they return to year after year. And then in around November, December, they'll leave and find a hibernaculum, which is a place that they'll go to hibernate into the spring. Could be a man-made structure. It could be a cave. In Baltimore, it's usually like a man-made structure. And then once spring comes back around in March, they usually reemerge, and then the cycle starts again. So from previous research, there are 6 bats that have been studied before, and that is the big brown bat, the little brown bat, the hoary bat, which is the bat that we see right here on the right, which is actually my favorite bat. I think it's a really cool looking beautiful species that we have around. And then we have the tricolored, the silver-haired bat, and the eastern red bat. The thing about these bats is the way that we've surveyed them to see which bats are around is through acoustic monitoring. And I'll get into a little bit about that. But that basically use the acoustics, the echolocation on the bats to see what species are around, what species are calling, and then that's how you can identify them. Now, bats are a big part of our ecosystem. They provide many ecosystem services. 10 bats have been identified in Maryland, but again, those 6 that I've mentioned before, those are the 6 that have been seen in Baltimore. So a critical part of ecosystem services for bats is their role in nutrient cycling, pest control, which is a very, very big one, huge for agriculture, and even pollination. Some species, as they go down to capture like an insect that they might be following, they might get some pollen on them, and then they might go to another flower or plant and then do it again, and that might pollinate a plant. So pest control and pollination are big ones. I have this fact here that 70% of bats are insectivores, and USGS believes that over $1 billion annually is saved in pest control from crop damage just because of bats. So they're this weird creature that comes out at night, but they're like a very big, important part of our ecosystem, as well as the ecosystem services they provide. So very valuable. So in recent years, I'm sure you guys have but there are several conservation challenges to protecting and saving the bats. Habitat loss and climate change and disease are the big 3 that come to mind. So I'm sure some of you have heard about white nose syndrome. I think I'll talk about it a little bit more, but it has decimated a lot of the populations of bats that we see today. So almost 90%, if not over 90%, I think the northern long-eared bat, I just read that 97% of its population pre-white nose is now gone. So it's down to the last 2 or 3%. White nose syndrome has been very, very destructive to bat colonies and their populations. Bats take a long time to recover because they only have 1 or 2 pups at a time. So if this disease is wiping out so many of them at a time, they can't really replenish their population. And then the northern long-eared bat was officially listed as endangered last year around this time. I think last November it was officially listed. It's actually a species that I'm working with now at Virginia Tech, which is kind of sad. It's fun, but it's really sad to see this beautiful species just kind of slowly go extinct. Also, another conservation challenge that I didn't really think of, but working in an urban area and talking to a lot of people, it comes to mind. Just public perception of bats is kind of like they're this weird creature that comes out at night. They have disease, they bite you. So that can kind of make it difficult when you're trying to come with a message of, no, these creatures are important. We need to protect them. So those are just some of the conservation challenges that I see. So in urban areas in particular, finding bats in a city like Baltimore can be very difficult. They're very small. They hide in very small nook and crannies of old abandoned buildings. So it takes a lot of effort to find them. Again, general awareness just about bats is difficult to come by. Not a lot of people like them. Not a lot of people want to do much for them. So it can be kind of difficult to get the word out. And then finally, there's not a lot of research previously about bats. So it's kind of going in blind. At least before, with other species, you can look through the literature and see what has been done to kind of help you, but it's not much of it in urban bat populations. So it's a little bit more difficult to find what you need to find for bats. So now I'm going to talk a little bit more about my method. So I said that it was non-invasive, and that's why I have this picture on top, which is me holding a vial of guano. So I guess my first step was I wanted to find where the bats were roosting in and around Baltimore City. So a lot of these likely spots were homes or attics or buildings, even bridges. And I looked through as many as I could. I tried to also put up bat boxes as a way to bring bats to certain areas so I would know where I could find bat guano at. I even used people that contacted people that use apps like, I can't remember the name of it. The apps where you can identify wildlife species. I would reach out to people that had saw bats, and then from there, they would tell me where they saw it, and then that would give me a clue on where I can look to find bats roosting at. Acoustic monitors were a big part of my process as well. So I could put up an acoustic monitor, which I'll show in a little bit. that tells me where the bats were calling. So there's a likely chance that the bats are at least staying somewhere nearby. And then I can limit my search area to there. And again, establish some bat roosting sites by installing bat boxes. A lot of them weren't taken up on, but at least they're there. So in future years, they can be. And some of them weren't taken up on. So I thought it was a great method of getting people involved by providing bat boxes as well as helping the bats as well. So for guano sampling, the idea is that these bats would go out and they would eat a bunch of insects. And then those insects would come from contaminated water and soil sources. And then the bats would eat a bunch of them, and those contaminants would then move from the insects to the bats. And then they would begin to bioaccumulate and biomagnify in the bats. They would then excrete it as guano. And then if I found out where they were roosting, I can then go out and actually find the guano. So that was the method. And I wanted it to be as non-invasive as possible because I didn't want to hurt the species that's already on the decline. You can do hair samples, you can do wing punctures. I did try to do the hair samples, but I just didn't have enough bats at the time. Wing punctures have been done. If you're able to catch the bats, at the time when I was working in Maryland, they didn't want us catching any bats because they didn't want to spread white nose. And I think there was also some precautions because of COVID. So I went with the next best thing, which was the guano. And in research that has used guano, it gives the same kind of measurements as if I took an organ sample. So it was comparable. So it seemed like the best way to go about it. A lot of the samples, or at least the methods that have been done before, they usually take an organ sample, so liver, kidneys, or a carcass if they can get their hands on it. But in general, I think that it's better if you can go non-invasive if you're getting the same amount of data anyway, especially for a species that's threatened or endangered, like some of the bat species I was working with. Again, so bat guano has been sampled before in bats in 65 different species, but only 4 of those species have done multiple times. I think the big brown bat is one of those species. I think it's big brown, and I forget the other ones. But cadmium, chromium, copper, and lead have also been measured in bats. So arsenic and mercury, which I was using for my study, was going to be unique. So I thought it was a good way to build upon the foundation of knowledge that we already have about bats and environmental contaminants. This also gave me an opportunity to connect with a bunch of different organizations, such as I'm speaking with today. So I got again, I'm not from Baltimore, so I wanted some kind of people from the community to guide me on what best to do. So I reached out to urban gardens, urban farms, museums, anyone in Baltimore that can kind of guide me on the issue and get their perspective on it. So Baltimore Green Space, BMI, Bliss Meadows, the Baltimore Birding Club, all these groups are groups that I worked with in some way to either set up bat boxes, have presentations, or have like evening bat walks, which is a way to get us to go out, walk out at night, and then get a active count of bats that are flying in the area. And then with this network, I was able to not only better figure out where the bats are staying and where I can get guano, but I could also get people engaged and involved in the process. Also, citizen science was something that was pretty interested in for this project. And basically what it is just having people from the community, citizens come out. Even though they're not experts, they can come and help collect, and they can come sample data. When you think of science in the past, it was an amateur field. Amateurs would go out and it was their hobby to do science, but we kind of moved away from it, and now we see it as more of a professional thing. But I wanted to bring it back and get people involved and engaged and have experts come out and provide their expertise. Some of them knew more than me, so it was really cool to have. It's been growing in the field of zoology and medicine and public health. I see so many cool projects online or out that involve citizen science. I think that ecology and conservation is a great place for it to go. So that's why I wanted to include more about it, and I'll talk more about that. So for citizen science, there's programs like Universe. I think I used SciStarter for mine, and I named the project Rhythms of the Night. Thought it was a catchy name to get people interested. And what it does is it trained participants to come out and identify bat species based on echolocation. And it kind of looks like this, and I'm going to show more about bat calls in a second. But what it did is I would get bat calls on my acoustic monitors, and I can put them on the website, and then anyone that was interested was able to come out and then help me identify what kind of calls they had based on the training program I had. So it helped me go through a lot of my data a lot quicker, but it also got people engaged in the process, which I thought was a lot of fun. So here's me finding a tree to put an acoustic monitor on. So that little box has a microphone, and it kind of looks like a trail camera, but it's a little different. It just uses the acoustics of the bats. So the bats put out their echolocation when they're foraging or moving through an ecosystem. This has a ultrasonic microphone that would pick it up. I usually set it up for about 3 to 4 nights at a time. Set it up for just the evening, so maybe at 7 at night to 5 in the morning when the bats would be active. I usually like to set it up in an open area, facing like a field or an open spot where like insects would be, so that one, I would get calls of bats as they move through the area, and two, so the echolocation wouldn't, if you put it in a forest, the sound waves can bounce off trees and get kind of muffled or distorted. So I wanted it to be as clear as possible. So this is me setting it up, pretty simple, and I just have it up there for a couple nights, and then I could review it at a later time, and it will show me exactly what bats we have or what species. I do have to manually go through and look at it, but each bat has its own identifiable call. So that allowed me to do it when I wasn't around. There are active monitors, which is like the one here. So that's in the cell phone. It has a little box on the top. Same thing, it's in an ultrasonic microphone. And in real time, you can go out and you can listen to the bats and see what bats are flying overhead. And if you have it set up in the right way, it'll tell you which species it is and it'll show you the call. This is great for people that just want to have a hobby or kids that just want to have fun at night, but it also has real implications for research, which is how I used it. Again, I used it for a lot of bat walks around the city. Just hand those out to people in the crowd. They set it up in their phone because it's a free app, and then we can just walk around and listen to what species we have, which is a lot of fun. So here's a little bit more about, I guess, the technical side of the call identification. So once I actually had the call in hand, it would look more like this down here with a bunch of clicks. I think they're actually really beautiful. The parts with the bright yellow or the orange is where there's like more power in the call. And each call or each wave has its own signature to a bat. And at different parts of a bat's foraging or activity in the landscape, they have like a different call. So here you have like an approach phase, and then as them putting out a call, seeing who's around. And then at some point there'll be a feeding buzz, and then they'll listen in to see if there's any response or if they're getting any kind of call back from the echolocation. And then once they were in, they can approach, and then they can actually capture, and then they move on to the next. So you see it's approach, they're putting out like a very broad call, and then they get faster and faster as they actually find something that they want to zoom in on. And then it gets faster until they capture their prey, which is usually down here, and then they cancel it for a little bit, and then they move on to the next. And then when you're looking at these calls, you want to know what species are local. So my identifier had all 10 of those species that were in Maryland, because I was in Maryland, even though in Baltimore, I didn't expect to see them all. And then you just go through a checklist of what the minimum and maximum frequency is, how long each pulse is, and then if there's bounce. And what bounce is if the calls are steady at the same frequency, or do they go up and down? And then just the total number of calls and then the shape of it. So this call here is a big brown bat call. They have like a nice swoosh to it that gets quicker, and then it cancels off a little bit. So this, I hope, is not too loud. I do have a couple of calls for you guys to listen to of 2 species. Let's see here. So that's one. And that's the second one. And then with that information that we used on the last slide, we would tell that this one, the first one up top, is a big brown bat call. And then the second one is the Eastern Red call. Both really cool calls and very common in Baltimore. So a lot of fun to capture those. And then again, so this is what it looks like when you get it on the computer. Those colors show the power at different stages that the bat is actually putting into a call to send out to see who's around and if there's any insects. And then again, the frequency, duration, all of that matters, and the shape can give you clues to identify. It's not always perfect. There is some overlap. So big brown bat and the silverhead bat have very similar calls. So it does take some training and some time. But these are all clues that you can use to identify bat calls as you're going through so much data. Bats put out a lot of calls, so it's a lot of data to go through, but you definitely get good at it. So to give you guys a little scope or idea of the scope of this project, so I had total 41 different sites all around Baltimore. So the blue sites that you have up here and down here, those are sites that I had bat boxes up, but I didn't get any guano, but I also had acoustic data up and I was able to collect. So normally I would put up a bat bot or a bat monitor at like a person's house or at one of those parks I said, and then I would wait to see if I got any calls. And if I did get a bunch of different calls, I would figure it'd be a good place to first look for guano and also set up a bat box in case we got lucky. 17 of these sites, the red ones here, are places that I did the same process, and I was able to like put up a bat box, or I was able to find a roosting location. So down here at Patapsco, I found a bunch. I found some at Patapsco over here. I think that was a silver-haired bat, which was really cool, and then some scattered spots around. And then I had some other sites, the green, that I put up acoustic monitors. I got no calls. I got no box installed, and then I didn't find a roost. So those were spots I kind of like were duds. But they were still cool because maybe next year, maybe someone else will pick it up and find some bats there. So I had 7 sites that provided guano within the city, without outside of the city, and then 10 other sites in the city limits that had guano. So with the acoustic monitors I used, again, I would set those up for about 3-4 nights at a site. Sometimes it went longer if I just couldn't get back to my monitor in a rapid time. Usually from like 8 p.m. to 5 a.m. I have it going because that's when it started getting really active. I would say most of my calls around 9:30 to 11 is when you get the majority of your calls. That's when they're extremely active. Of the 6 species that I identified before, 4 of them I had calls captured of. So I had big brown, I had a hoary bat, I had eastern red calls, and I had silver-haired bat, which means I didn't have tricolored calls and I didn't have little brown calls, which are 2 species that are very much impacted by white nose syndrome. So that might be a clue in that their populations are declining and struggling in urban areas. One bat that I don't think was previously recorded, though, that I did capture on my monitors were evening bats. And this is an evening bat right up here. They're really cool. They usually end up in more swampy areas, so I'm not sure exactly, maybe somewhere in like Leakin Park or Druid Hill, but I don't really think those have any swamps, so not really sure why I caught one, but he was there. So I'm actually going to be working with them for my next project, so I'm excited to learn more about them. So also here's me putting up some bat boxes. I think this was like Green Street Academy in Baltimore City. I wanted to set up as many bat boxes in a way that people can be engaged. So I'd have like bat box building workshops where people can come, build a bat box and get one for free, and then they could take it home and set it up. And the reason for that is because, again, bats are like extremely affected by white nose syndrome and just urbanization. So providing as much habitat as possible, I thought was critical. So gave out a bunch of bat boxes, also with white nose syndrome, which complicates things, which you can see here for those that don't know. So what white nose syndrome is it's a fungus that grows in caves or places where bats are hibernating. It gives them this white fuzzy nose as the fungus grows, and it also gives them spots on their wings and spots on their body. What it does is it wakes them up out of hibernation during the winter. Bats, once they're awoken from hibernation, they need to eat. Their metabolism is fast, so they need to eat lots of insects. There's no insects in the winter when they're hibernating, so most of them usually starve or die from dehydration. For my research now at Virginia Tech, I am catching some bats that have survived white nose syndrome, which is, on some scale, it's a good thing because it means that in some ways they're finding ways to survive. But on the other hand, they get reinfected each year that they go back to the hibernaculum, so it might not mean so much after all. But yeah, so it's a very big problem for a lot of species. Species like the eastern red bat, which roost in foliage and trees, they don't actually come in contact with the disease, so they're okay. But species like the tricolored, silver-haired, big brown, little brown, northern long-eared, they all set up hibernaculums in colonies, in caves and places like that. So it spreads really quickly, and they all get infected with the disease. That's why their numbers are dwindling so much. So bat boxes can be a way to help like impact or fight against the disease and hopefully, give them some habitat that's not infected. And here's a little bit about those bat boxes that I was building, but I couldn't have done that without educational programming, which I thought was like really important. I wanted to get people involved and people interested in bats in an urban area. I think that was really cool. So I would have these workshops and I would provide the bat box, the kits. People could take them home, build them, do whatever they want with them. It was just a nice way of providing community stewardship and just engagement to get people involved with it. And with that, I also did outreach with about 20 different community organizations. This is me talking. This kid was so into it when I was just talking about like bat boxes and like acoustic monitors. He loved it. It was a lot of fun to do. So just going out, getting people interested, and if you can, dispel any kind of like notions or any anxiety against bats was a lot of fun of just kind of like switching it up and telling people, no, bats are like really cool and this is what we can do to protect them. And it gave me a big opportunity to connect with people all over the city and the county, which was a lot of fun. So in conclusion, I think I hit it, but like I think bats are really cool. I think they're a great part of the ecosystem. I think the services they provide are immense. As biomonitors, they provide a lot of information and context about the ecosystem and what heavy metal contaminations we can come in, but also provide a way. And I think as like conservationists and ecologists, we should think of ways to also get people from the community, from all over involved in the projects that we do. Thank you. And with that, any questions?
All right, there is a question from Valerie. Very interesting and well done. I learned a lot that will help me when I set up a bat box. Warm thanks and congratulations on your good work, Christopher.
Thank you. That's awesome.
I have a quick question. So, like, are we? I guess you said this, but maybe I'm confused. But so, are the heavy metal concentrations in bats causing problems for them? Like, do we know what's happening because of that?
Yeah, so in terms of problems, like there are behavioral problems that have been noticed in bats that are like overly contaminated at a certain threshold. We'll start to see mental issues, a lot of reproductive issues with bats. So that could be complicating more the things that we're seeing with survival from white nose syndrome. Again, there's so like little like limited research on bats in urban areas and in terms of being a biomonitor that I can't really say for sure. I can't really make the connection if like we're seeing decline based on certain like heavy metals. It was outside the scope of my research, but I would think that we would see at least something in terms of like behavior and reproduction, but I can't say for sure.
Gotcha.
Yeah.
Well, there's another question from Carol. Do you have any observations yet about the proportion of bat guano samples that have each of the heavy metals?
So I'll say, so I'm working on getting the chapter published that dealt with the contamination. I'll say that all 5 of those metals I saw in bat guano in different spots. So a place like Patapsco, I saw a pretty high level of arsenic. That might be, I know that arsenic is often used for treated wood, and a lot of the bats are using the buildings or the picnic pavilions there. So there might be some kind of contamination coming in from that. I will say a lot of locations that I sampled that were along the Gwen Falls also had like a lot of contamination as well of like cadmium and chromium. So it was a little outside of the scope of my project to see exactly. I wanted to do more research into where, like, once I found the contamination, where the contaminants were coming from. So maybe do some like kind of sampling with the bats and insects to get more definitive answer. I didn't get to do that because I moved from UMBC, but I think that'd be a really interesting question that somebody could pick up of now that we know where contaminants are, what contaminants we're seeing, and at what level we can better identify the sources of contamination. But yeah, so I would say mercury I didn't see as much of overall, but arsenic, cadmium, chromium, I saw high levels of in certain spots.
Gotcha. And with the bat boxes, did you learn anything about like where you can be more successful, like what positions or locations they seem to prefer?
So I know in terms of position, they like it at least all my boxes that I had like up 12 to 15 feet up, they seem to
Mhm.
like that. Also, if you're having faced south or east, that's best for thermal regulation of the bats as the sun moves. It gives them the right amount of heat so they're not too cold but not overcooked. In terms of what might be best suited for them, it was tough. Only because I think it has to do with what available habitat is around. So a lot of my parks and stuff that I put them at, some of my gardens, there might be better habitat for them than a bat box. So they might have
Mm,
used that. But in my more urban areas, so I had a couple of buildings that I put it on the side of that I got lucky and I got some bats to go into. So that might be, hey, there's not much available space for us. This might be a good spot. It
gotcha.
was, I would say, it might take a couple of seasons if you put up a bat box at your home before you see any activity. But yeah, it was hard to say for sure.
And I'm guessing shouldn't use treated wood that contains arsenic as bad box material, right?
No, no, yeah. If you can avoid that, please do.
Yeah, awesome. Any other questions? Uh, there is a question from Gene. I'm interested in documenting what bat species occur on our property near Loch Raven Reservoir. Can you recommend any equipment makes, models for active acoustic monitoring?
Yeah, so this right here is kind of that thing I showed in the slide. It's called the Echometer Touch too, and I can write that into the chat.
Yeah, that'll be great.
But it's about 150 bucks, I think. So it's a little expensive, but let me see if I can put it in. But once you, yeah, it's a free app. So once you have this piece, they have one for an iPhone, they have one for an Android. You can put it into the app or into the phone itself, download the app, and then this will tell you exactly what species. You do have to mess with the settings a little bit and just put in where you're located, so it knows what species to look for. But once you do that, yeah, it'll tell you exactly what species are around. Let me see if I can put this in chat. Yeah, the Echo Me to Touch too. I had a couple of those. I think those are the best product that you can have in terms of like going out that's not too expensive, but it's the right amount and you get great data from it. And it's a lot of fun. I let one of my advisors use one with her daughters and she loves it. So
Awesome. Well, there's another message from Marcia. I think you can read it yourself too, but I'll just say it for everybody. Go Blue Hens. Good to see another UDEL person out there conducting research.
yes, I love it. Yes. Had a great time at University of Delaware. So I love meeting up with fellow Blue Hens.
Yeah, where is University of Delaware again? Is it Wilmington or?
It's like right outside of Newark.
Oh, okay, gotcha.
Yeah. Yep.
Newark, Delaware. Gotcha. Yeah,
Newark, Delaware. Yep.
awesome. Anybody else have anything to ask or say? Okay. All right. Well, thank you so much. Thank you, Chris, for spending time with our group and helping us understand what you've been doing at UMBC and continuing to do at Virginia Tech. Good luck on your research, and I hope we can circle back in a couple of years with some more findings on our bat neighbors.
Awesome. Yeah, that'd be great. If anyone, my new research is going to be mostly in Rock Creek Park in DC, so not far from
Awesome.
Baltimore. So if anyone sees any monitors or bat
Mhm.
boxes or sees me out there, please say hi. And yeah, hopefully I
Awesome.
found something good.
Great. Thank you all.